But before we begin, I would like to ask you all to bring out your cell phones and make sure that they are turned off. Thank you for your cooperation. Now let me introduce Professor Urban Lendahl, Chair of the Nobel Committee and Professor at the Department of Cell and Molecular Biology here at Karolinska Institutet. And Professor Lendahl will introduce the Nobel laureates and give the motivation for the prize. Thank you. Please.
Dear laureates, distinguished guests, friends and colleagues, I'm privileged and honored to stand here today to introduce the two Nobel laureates in Physiology of Medicine 2012, Drs. John Gurdon and Shinya Yamanaka. Together, these two scientists have fundamentally changed our view of the cellular differentiation process and how we view the specialized cells in our bodies. They've shown that the differentiated cell state was not cast in stone, but that a dormant potential of rejuvenation could in fact be unchained in these cells. This has not only made us view cells in a new light, but also open new avenues to gain insights into disease and in the future to improve diagnosis and hopefully develop new therapies to hitherto incurable diseases. Their achievements also provide a prime example of how fundamental discoveries rooted in basic science can profoundly impact on, or tempting to say, rejuvenate many areas of medical research. To put their discoveries in context, let us briefly revisit how we came to learn about differentiated cells. The journey from a fertilized egg to an adult individual has always fascinated mankind. And an early account on developmental processes dates back to Aristotle, when he temporarily left philosophy to go to the Greek island of Lesbos and together with Theophrastus ponder on botany, zoology, and generally on the wonders of life. For a long time, then, progress was more limited. But with the advent of the first microscopes in the 1600s, this allowed objects in the size of a cell actually to be observed. And Robert Hooke, one of the pioneers, he looked at a section cut out from a piece of cork, and he observed structures that he thought resembled miniature versions of a monk's chamber, and hence he called these structures cells. And the modern cell theory and the concept that the cell is the building block of all living forms that was established in the 1800s by scientists like Theodor Schwann, Matthias Schleiden, and Rudolf Virchow. And as histology evolved and microscopes and staining techniques got better, it gradually became clear that our bodies contained many different types of specialized cells, each with a unique morphology. And here Ramon Cajal's beautiful drawings of cells in the nervous system comes to mind. In the light of this, the notion that a differentiated cell was an endpoint, the final stop on a developmental journey, grew stronger. And this notion was reinforced in the early days of cell and molecular biology, but that the different cell types not only looked different, they also were endowed with different compositions of proteins and later on also RNAs. Furthermore, with the discovery of mobile genetic elements by Barbara McClintock, it seemed conceivable that the genetic material in the cell would be irreversibly altered during cellular differentiation. There were also entropy-based models, often portrayed as a mountain landscape, where the differentiated cell state was conceived to be the most energetically stable state, and the differentiated cells would peacefully be resting in the mountain valleys. Put shortly, the dogma was building that the differentiated cells could not be persuaded to make a round trip back to their original immature state. And such was the scene when a young John Gurdon took on this question. Discouraged early on by a teacher to venture into science, but equipped with a sharp intellect, curiosity, and, I've been told, an aptitude for doing things on a small scale, such as building 3-masted sailing ships in walnut shells, John Gurdon asked if a differentiated cell nucleus could restart development if it was placed in the right environment, the enucleated egg cell. And this work, carried out in the frog Xenopus levis, began in the late 1950s and culminated in a landmark publication in 1962. And this discovery really ran counter to the prevailing view that the cell differentiation state was irreversible. But John Gurdon was right. And based on his findings, somatic cell nuclear transfer has since been expanded also to mammals, notably in the form of doll-eater sheep. John Gurdon originally trained with Michael Fishberg at the University of Oxford, where he received the PhD in 1960. After a stay at Caltech from 1960 to 1963, he returned to Oxford to then later move to Cambridge University in 1972. From 1989 and onwards, he's been with an institute in Cambridge that since 2004 carries his name, the Gurdon Institute. John Gurdon has received numerous awards, including the Wolf Prize in 1989 and the Lasker Medical Research Award in 2009. He was elected Fellow of the Royal Society in 1971 and knighted in 1995, so it should rightfully be Sir John. And with this as a background, please join me in welcoming John Gurdon, who will present his Nobel Lecture under the title, The Egg and the Nucleus, A Battle for Supremacy. John.
Should be all right, shouldn't it? So first, let me thank Urban Lendahl for his very fine introduction, and then say that this is, of course, a very special occasion for me, and I'm enormously grateful to the Committee for Physiology and Medicine for this unique and immense privilege of finding myself here today. So I've chosen to use this title for my talk because I think the interaction between egg and nucleus, leading eventually to reprogramming, can really be summarized as a battle between the egg and the nucleus. And at the moment, I would say the egg is doing very well, but the nucleus is slightly better. And we need to realize the full potential. We need to give the egg a little more help to try and reduce the remarkable resistance of the nucleus to being reprogrammed. So these are the subjects I will talk about. A bit of background, explain the history of the subject, and then talk about the egg, how it is trying to reprogram a nucleus, and then talk about the nucleus, how it's trying to defend itself by being reprogrammed, and finally a few words about prospects. So I always think one should start with a question, and in my case, the question was, all this time ago, whether all cells in the body have the same sets of genes. Simple enough question. The originators of this whole field were Robert Briggs and Thomas King, and they had great success in 1952 in succeeding for the first time in transplanting a somatic nucleus to an enucleated egg and getting normal development from it. However, when they tried to do the same experiment with the nucleus of a more specialized cell, or actually just an embryo, a little bit later on, they found that there was no successful normal development at all. By the time they took nuclei from the early embryo, there was no success in obtaining normal development. So, of course, that led to the idea that as cells differentiate, they might lose the capacity of their nucleus to be reprogrammed, might be lost irreversibly. Well, I then started work a few years later and was advised to work with the South African frog Xenopus laevis, which is this one, and I was lucky at an early stage to get an apparently normal frog from the nucleus of a reasonably advanced embryo. This is the first adult we had historically. It was a frog of good quality. It lived for about 20 years, and it had about 4,000 offspring. So it was an indication that you can transplant a nucleus without generating any adverse effects. Now, a key step in this history was my supervisor, Michael Fishberg, who, in an ingenious way, discovered a genetic marker which was essential for proving that a transplanted nucleus on its own will give rise to a normal embryo. And this nuclear marker involved counting nucleoli in a cell. You had to be able to distinguish between 1 and 2. And in these cells, there is one nucleolus in each of the nuclei, representing, as it turned out, the site of synthesis of ribosomal RNA. And in the wild type, which was called a 2-nucleolus strain, there are 2 nucleoli in nearly all cells. The 1-nucleolus mutant was a deletion of ribosomal genes, and it was immensely valuable so that one could mark the nuclei being transplanted and show that the resulting embryos always had 1 nucleolus in their nuclei and never had 2. So with this background, the Xenopus work led, like Briggs and King, the success rate went down the more advanced were the embryos from which nuclei were taken. Nevertheless, even when you took swimming larvae, their cells, to a significant extent, were able to produce entirely normal embryos. And that then reopened the question of whether, as cells differentiate, their nuclei might still be able to generate normal development. So looking again ahead a bit, we started using the intestine of a feeding larva, so its intestine cells were functional, and in this case we could use the GFP marker to mark these nuclei, transplant the nuclei to eggs, and when you do that, you sometimes get normal embryos, but more often you get these partial embryos with normal cells at one end and a complete failure of development at the other end of the embryo. Now, these embryos will die. However, they die within about a day. However, it's possible to take these cells and retransplant a nucleus from those into another set of eggs, many of which will then develop normally without this making a defective embryo. It's also possible to take a few of these cells, which you may remember are GFP marked, and transplant them as a graft to a normal host embryo like this, and you can see the graft stuck onto the normal embryo, and when these develop, they can form entirely normal embryos, and all of these green cells, each one is a single muscle cell, nicely orientated with respect to the host cells, which do not carry the marker, they are black, and it shows that the functional muscle can readily be derived from the nucleus of an intestine cell. So that was the origin of much of this work. And when we summarize that, put that together, we can add up the success rate of the so-called first nuclear transfers and add to that results obtained by the serial second nuclear transfers, and also include grafts made in the way that I've described. And when you do that, the success rate with which you can obtain, for example, functional muscle and nerve from intestine is around 30%. So that says that the cytoplasm is pretty good at redirecting the nucleus of a muscle cell so that instead of continuing its life as intestine, it can, the cytoplasm of the egg can switch it back to the beginning of life and send it out in the direction of muscle and nerve. But at this point, this success rate of 30% also means that 70% of the embryos do not achieve that. And so in terms of this conflict or battle, we would say the nucleus on the whole is winning. Cytoplasm does pretty well, but most of the nuclei are unwilling to be changed back to redirect their differentiation. So at this point, I should acknowledge the work of Wilmot and Keith Campbell, who in 1997 were able to do nuclear transfers in the lamb, in sheep. An interesting question, of course, is why there was a gap of some 40 years or so between the early frog experiments and the sheep experiments. And the short answer is that the people working with mammals, mostly with mice early on, did not actually follow exactly what was done with the frogs. For some reason, they chose to use fertilized eggs, which were extremely difficult to deal with. And Ian Wilmot and Keith Campbell chose to do exactly what had worked with frogs and obtained these wonderful results with sheep. So, in summary then, the results with the frog and mammals are remarkably similar. In each case, you get about a 30% success rate. If you take mammals reaching birth or Xenopus reaching tadpole stages, and as you take nuclei from more advanced stages, the success rate goes right down. So that's a measure, again, of this resistance of the nuclei defending themselves against the onslaught of the egg trying to make them change direction. Now, I'll now refer to a remarkable experiment of James Byrne, who was a student of mine, but he didn't do this work with us. He did it later. So he took monkeys, skin, and eggs from another monkey, did the nuclear transfer, or cloning as it's called, made the embryonic stem cells, brilliant discovery by Martin Evans, and was then able to increase the cell number and add factors to make these cells now turn into a beating heart. And if I am patient, this will probably show the beating heart cells from the monkey skin. It's a sheet of these thousands of cells beating remarkably well in these cardiomyocytes. Of course, the problem is when you transplant those cells to a heart, they do not, in present technology, integrate well. But that was a very elegant demonstration of what can be done. Now, briefly, let me mention this phenomenon of epigenetic memory, which in effect shows that a transplanted nucleus will very often remember where it came from, even though the egg is making its switch fate. And in this case, one was taking a muscle cell, transplanting its nucleus and make an embryo, and then separate the embryo into the future neuroectoderm, or nerve and skin cells, take away the future muscle bit and separate the endoderm bit, and rather amazingly, the future nerve and skin cells, or gut cells, continue to express muscle genes to a surprisingly large extent. Start with muscle, go through and go into the nerve lineage, but nevertheless, those cells remember somehow that they came from muscle. We had reason to think this might have to do with a particular protein called histone H3.3, and so we were able to use a mutation in that gene to essentially compete out the normal histone H3.3, which is present in the egg, and show that when you did this, you eliminated this memory. Instead of being 50% or so, it went down to just a few percent. So that indicated that histone H3.3 is one of the factors the eggs use to increase transcription. And when you transplant a nucleus that is already committed to one fate, the egg enforces continuation of that expression, which makes it very hard to reprogram it. So this is another reflection of the amazing defense that the nucleus of a specialized cell will put up, as it were, to resist the reprogramming activities of the egg. So in summary, then, the epigenetic memory, as we call it, reflects the activity of this variant histonate 3.3, which promotes transcription of active genes, and the egg cytoplasm can reverse that, but only with about 30 to 50% efficiency. So that then makes one turn to how one can start to analyze this conflict of interest a bit further. And I'm now going to talk about these cells called first meiotic prophase oocytes, which are the progenitors of the egg. Now, this simplifies the analysis to a considerable extent, because in the experiments that I've just been talking about, you transplant a nucleus and you get an embryo. But before that happens, there are numerous cell divisions and replications before the embryo can form an adult frog. And this is where the reprogramming happens, where the egg is switching the nucleus back. Now that complicates analysis very considerably, because when this happens, there is incomplete DNA replication, and this damages the somatic nuclei so that many of them are no longer capable of following normal development. To overcome that, one can use these egg progenitors, which we call oocytes. They contain a large object called the germinal vesicle, and one can inject multiple nuclei into this, and this oocyte will continue life. It has no DNA replication occurs, but intense transcription. And after a couple of days or so, these nuclei have now switched. They have now been reversed, rejuvenated, and made to express embryo genes. So you don't get new cells, but you've done a large part of the reversal process. We can understand this by realizing that the egg progenitor or oocyte starts life as a germ cell, and then it gradually forms the mature oocyte or egg. During this time, its nucleus builds up a large reserve of these reprogramming factors. And only when the oocyte eventually becomes an egg, in what seems like a flash, just a moment, the egg will form and will be fertilized normally, make cells, and already you start getting different cell types, the red, blue, and yellow. So the oocyte is preparing itself for a long period of time, like 100 days, building up these reprogramming factors ready so that when fertilization or nuclear transfer happens, it can immediately embark on normal development. That's why the oocyte is such a valuable source of these reprogramming factors. The oocyte looks like this for the frog. It's a very big cell, about 1 millimeter in diameter, and its nucleus, the germinal vesicle, is also very large, and the germinal vesicle contains this reserve of reprogramming components indicated in red, and these are distributed to the egg when the oocyte changes into an egg, and then to the embryo, and they are necessary for normal development. So these are the key reprogramming factors that are needed to switch the somatic cell backwards in development. And if you look at gene expression, this works fine. So here are these nuclei, which are adult nuclei, and mammal ones work fine. If you use rapidly dividing cells like HeLa, within a day, they have already started expressing the key pluripotency factors, which we'll hear more about later. On the other hand, when you use nuclei specialized cells like mouse thymus, they are significantly resistant. They resist the reprogramming factors of the oocyte. So this system enables us to begin to analyze what is really happening. So this led to the oocyte transcription assay, as I call it, and the major features of this, which are turning out to be very useful, is that you can transplant many nuclei, several hundred, makes analysis easier. The new transcripts, the pluripotency gene transcripts, accumulate nicely with time. Most notably, the genes that are being activated undergo recurrent transcription continually, many hundred times a day, and that cannot be done in other systems. And lastly, these injections into oocytes show this resistance or defense that we're interested in. So now I turn to the transcriptional activation. In other words, what is it that the egg is doing that is somehow able, to a fair extent, to rejuvenate adult nuclei and send them back to the beginning of development? So I see it as the egg attacking this somatic nucleus, trying to force it backwards. And so how does this happen? In normal life, the sperm is a tiny cell, and in mammals it forms this enormous male pronucleus. It undergoes a massive expansion because the egg forces that. And so indeed, when you transplant nuclei, somatic nuclei, that happens also. Here are these few hundred transplanted nuclei, and in a short while they've formed these balloons, big vesicles, with a huge decondensation or opening up of the genome. So the genes are now accessible to factors from the egg. And part of this process is seen by the uptake of special molecules of the egg, which in this experiment have been marked in red. And we start by transplanting nuclei in which the adult type linker histones are shown in green. And when you follow this process in time, one can see that the green molecules disappear and the oocyte's own red molecules take over to a remarkable extent. Almost all these transplanted nuclei have taken up the red special histone, linker histone of the oocyte. The few that haven't, the green ones, are interesting. They are ones where, when we transplanted a nucleus, by mistake, we transplanted a whole cell. If you transplant a whole cell, nothing can get into it. You have to transplant the nucleus, not the cell, for the egg's components to gain access to that nucleus. And this process happens surprisingly quickly. This is in about 2 or 3 hours. This whole process has happened, and that's amazingly fast for a somatic nucleus that might expect to spend the rest of its life as a specialized cell. Within this short time, it can be the whole reversal process can begin due to these special histones contained in the oocyte. And soon after that, there's another chromosomal protein called histone H3.3, which we've mentioned, also invades the incoming nuclei. And one can now follow this in some detail by looking at the different forms of RNA polymerase, which also enters these nuclei as well. So these are the transplanted nuclei, 24 hours and 48 hours after transfer, and even the elongating polymerase II is successful in gaining access to these transplanted nuclei. At the same time, the nuclei lose some of their components. The RNA polymerase brought in by the donor nuclei disappears, and the tartar-binding protein of somatic cells disappears and is replaced by the tartar-binding protein of the oocytes. So these are the switches which are happening very quickly, and they arise largely because these components are so abundant in the oocyte that they force their way into the nucleus just by mass action. Such a huge abundance, they invade the nucleus and fill it up with these components. And all these components are necessary for the reprogramming. Here you can see that the different forms of RNA polymerase II follow each other within a short time and rush into the nucleus to start transcribing the genes which have been opened up by the special histone that we just mentioned. Another interesting thing is that the RNA polymerase which is needed to read these genes, transcribe these genes, has to come from the oocyte. So you can do an experiment with alpha-amanitin, which kills the oocyte's polymerase II, and if you use nuclei which are resistant to this drug, they, shown in the blue, are quite unable to be reprogrammed or gene switched on, but the oocyte polymerase if it's killed, will not allow that. Normally it allows it, as shown by the substantial increase in expression of this gene. So an important question is, where does the selectivity arise? So why is it that the oocyte or egg can switch some genes on and others not? And this depends entirely on the cell type, the original state of the genes in the nuclei being introduced. And it shows here that a gene which is very easily activated, called SOX2, transcription start site, that gene will take up the special histone, the polymerase, and it will be transcribed well. Now, globin genes are not able to be reprogrammed by the oocyte. They're resistant to it. They defend themselves against this egg activities. And nevertheless, they can't help absorbing this special linker histone which opens everything up. On the other hand, they're completely unable to respond to the polymerase, and so they are not expressed. So it illustrates the actual level at which this resistance or defense is operating. You can open genes up, but the actual machinery needed to read the genes cannot work if the nucleus from the adult cell is exerting its defense, so it will not allow these oocyte factors to operate. And so we can begin to put together these events which are needed for the oocyte to have its reprogramming activity. And these are some of them. There's no doubt a few others, but it gives us an indication of how the oocyte or egg attacks the somatic nucleus by overloading it with these abundant components which enter the nucleus and try to reverse it, try to get it to go backwards in development. It's quite useful to know what these are, because they are necessary. Without them, there would be no rejuvenation or reversal, and these are some of the components which are needed for this special process. So now we turn to resistance, or as I call it, defense by the nucleus. And I would argue that this is, in many ways, the most important part of the whole process of reprogramming. Why is it that the nucleus is able to defend itself against this enormous influx of components offered by the egg? And in the work that we'll shortly hear about, the famous IPS work, the success rate of reversal is normally very low. Originally, it was maybe 1 in 1,000 or 10,000 cells. So most cells have an extraordinary defense, resistance to reprogramming. And I would argue that if we could really understand that, we could assist the egg or other components in rejuvenating nuclei and make the whole process of reprogramming considerably more efficient. So what is the basis of this resistance? How do nuclei defend themselves against the reprogramming components of the egg? In one case, we know something about this, and you will know that in female mammals, one of the 2 X chromosomes has no gene activity. The genes are all repressed. They call it X inactive. Now, this turns out to be true if you take nuclei from adult cells, like mouse embryonic fibroblasts, they make no response, even in 3 days, to reprogramming by the egg. On the other hand, similarly inactive X chromosomes from the epiblast, it's an earlier stage of embryos, are very well reprogrammed. So the two states of this inactive X illustrate the difference between them, and hence tell us something about the component that is responsible for this resistant state of the adult nuclei. Now, the short answer to this is that it's been possible to identify this component, and it's called macro H2A, and you can do experiments where you eliminate that resistance factor, and when you do that, you then turn on pluripotency genes, OCT and SOX, even in the adult cells, very well. So this macro H2A helps to explain the resistance to reprogramming. It's one of the molecules which has to be eliminated from nuclei to enable them to be reprogrammed. So in conclusion, this macro H2A is a good example of a molecule that marks embryonic differentiation, and it acts as an epigenetic resistance factor to nuclear reprogramming. So that leads us to now ask, well, what kind of reprogramming is this? Do we see most genes in the genome being reprogrammed, or is that a rare and specialized situation? And when you analyze all the genes in the genome, you find that the reprogramming or gene selection 48 hours after nuclear transfer is highly selective. So we can see that genes which were off in the donor nuclei, shown here low level or nil level of expression, about half of them just stay off. A few more were expressed at a high rate, and they stay on after nuclear transfer. And a few others, when they're after nuclear transfer, they go from an on state to an off state, and a smaller number, less than 10%, are actually activated, and those include the pluripotency genes. So it means that the oocyte is not making a universal activation of all genes. It's quite selective. It will choose the kind of genes it wants to be reactivated. A useful guide to the level at which this reprogramming happens. So in conclusion, the resistance or defense of the nucleus is both gene-specific and cell type-specific. So there are two ways forward at this point. One is to start analyzing the chromatin, the molecules that sit on genes and control their activity, and we'll talk about that first. So the way forward here is to change these modifications. Genes have histones, and the histones have components on them which regulate the expression of the genes, and so it's possible to inject messenger RNAs on first day, and then you transplant nuclei on the second day, and then re-isolate the transplanted nuclei on day 3 and analyze what has happened. And when you overexpress by message injection these different demethylases or deubiquitinases, it has a substantial effect. It's possible to really remove these components on the transplanted nuclei and so see what effect they have. And this shows that this is quite effective. So normally you have high level of methylation on these nuclei. If you overexpress the demethylase, you essentially remove all those. So that's a good way of changing these components and hence determining what effect they have. As a matter of fact, it takes me back to one of my few experiments that I really found worked amazingly well. When a long time ago, I wanted to try injecting messenger RNA into eggs, and it's a project which would never have had any grant support at all, because it was known that if you take eggs, they are full of ribonuclease, absolutely stuffed full. And if you add ribonuclease to message, it destroys it immediately. Nevertheless, it was the kind of thing you think, well, we better just try it. So we injected this precious message, which had been exhaustively purified by people, into the egg, and amazingly, it worked perfectly. So why should that be? And I suppose the lesson you learn is that when you inject something into an egg, you do it almost no damage at all. You don't break open all these nuclease-containing components. And message injection has turned out to be an extraordinarily useful technique. And I reflect on the fact that if I had not had just enough funding to try it anyway, the experiment would probably never have been done. And so how lucky it is when you sometimes have a little bit of extra cash to try something a little exotic. So when you do these experiments, they are quite effective. You can see that here we have the heterochromatin 1, which is in nuclei. When you demethylate the lysine, they completely disappear. And as a control, this doesn't work with another particular form of heterochromatin 1. And this approach is proving useful because it's now we've begun to identify some of the components that actually create resistance. And there's a particular enzyme called D-ubiquitinase, which takes the ubiquitin away from histones. And you can see that when you do this, at time 0, there is no increase in gene activity. 2 days later, it's very strongly activated, and that doesn't work if you use demethylase. So we're beginning to identify those components on the chromosomal proteins that confer resistance or defense on the nuclei. And I'm sure there's a great deal of more work to be done in this area, but it's one way forward, and it's really only possible to do that if you have these very favorable cells which can be injected with almost anything you like. So that begins to answer this question, but it doesn't entirely answer the whole problem, because it turns out that even if you remove almost all proteins from a nucleus, from the chromosomes of a nucleus, nevertheless, the nucleus still has defense. So that's rather amazing. And one would say, well, in that case, how sure are you that this defense isn't something like the methylation of DNA, well known to prevent genes being reprogrammed? And that's easily disproved in many cases because you can remove all of the proteins from a nucleus, every single one, but leaving, of course, the methylation of DNA. And when you do that, the defense disappears. So you've taken something away which was creating this defense. So what could that be? And the problem then is to find a way of removing components progressively from a nucleus while you can still keep it alive enough to be transplanted. And this was done finally by finding a way of enwrapping the nuclei in a kind of cage so that you can take molecules away from them, and nevertheless, they do not explode and they can be injected. And the first obvious test here is to ask about the non-coding RNA. Is that what it could be that gives this defense? What you can do is to remove almost all of the RNA by digesting it away down to about 5 orders of magnitude, 100,000 times less RNA. Take it away, wash it all, add RNA back, and show the RNA is still functional. But so then you have RNA-depleted nuclei, and when you do this, you find that it makes no difference. So here we are with OCT4 gene or SOX2 gene, and you take away the RNA or you don't, you get exactly the same resistance to reprogramming because the slow rate it goes up. So that told us that conveniently you can more or less eliminate all of the non-coding RNA as a defense mechanism of the nucleus. What about proteins? Well, you can remove almost all the proteins by washing the nuclei in high salt and in Triton, and here they are almost all gone. And yet, when you've done that, you find that all of the most obvious proteins that could give defense to a nucleus, and there's a list of them here, have been removed, either completely or almost completely. Nevertheless, the resistance persists. This bar shows that unlike normal ES cells, which respond very well, these ones don't. And so there is something sticking to those genes which we don't know about, and which constitutes the ultimate level of defense against reprogramming. And that's the direction which this work is now taking. So I'll leave that point there and in a way summarize the point of this talk, which is to say the egg is designed to transform the sperm, which normally enters it at fertilization, into an embryo-active nucleus. And the egg is amazingly good at that. It does it in 99% of cases. The egg will transform this minute, completely switched-off sperm into complete activity. So that's what the egg is specially designed to do, and it's pretty good at doing it. On the other hand, it has a struggle. When you give it the nucleus of a specialized cell, it does a pretty good job, about 30% success rate, but it can't do the job completely. On the other hand, the nucleus of a specialized cell is designed for a different purpose. It's designed to keep everything very stable. And you will know that we don't have brain cells in our liver, or we don't have skin cells in our muscle. So once cells embark on a pathway, they remain very stable. And that's really why the nucleus builds up this defense against change. Of course, if the defense is completely broken down, there's a problem of cancer and other things. So the nucleus is designed to maintain its pattern of expression and try to resist any change. Now, the way forward from there will, of course, ultimately be, I think, to identify nuclear defense components, remove them, and try to facilitate or make more efficient this reversal or rejuvenation of the nucleus back to an embryonic state. But that will take time, and I'm sure the more immediate advance will be through these brilliant methods that Shinya Yamanaka has developed, which we'll hear about shortly. So that's, I think, where that is. And as I say, the prospect must be for us to try to defeat resistance and win efficient cell replacement. So I'll just acknowledge the people who've been helping me in this current work, and I would like to take the opportunity of mentioning a number of those who, throughout my career, have given enormous help and worked with me. Donald Brown was one of my first colleagues, and Ron Lasky has been with me for something, in one way or another, something like 35 years, helping and guiding all the way along. Doug Melton, Eddie Robertis, Lawrence Korn, Marv Wickens, Alan Coleman, Chris Graham and John Nolan were all early colleagues with me at the time the early experiments were done. And particularly I'd like to thank two people who started life as a technician. When I had no colleagues, no graduate students at all, had one technician, and these are the two ladies who used to relentlessly collect eggs and ultraviolet irradiate them and hand them to me for nuclear transfer. And it's a special pleasure that they are with us here. And the presentation today has particularly centered on work done by James Byrne and Stina Simonsen and Caroline Astrand, who are both from this country. Thank you.
1962 was a remarkable year. Sorry. Not only for John Gurdon's landmark paper, but the world also witnessed the birth of Shinya Yamanaka. And equipped with an MD from Kobe University and a PhD from Osaka City University, Shinya Yamanaka first considered a career as an orthopedic surgeon. But a postdoctoral stay at the Gladstone Institute in San Francisco opened his eyes for the world of cell and molecular biology. And after returning to Japan and the Nara University, he became intrigued by stem cells, and in particular by embryonic stem cells that were discovered by Martin Evans, who is sitting here in the audience today. Because embryonic stem cells can maintain their immature state and pluripotency during extensive in vitro culturing. And Shinya Yamanaka was interested in unraveling the basis for how this pluripotency was retained. And he made very important discoveries and contributed to deciphering the underpinning transcriptional circuitry, transcriptional factor circuitry, for example, by cloning the gene NANOG. However, even bolder, more courageous and far-reaching ideas were entertained by Shinya Yamanaka. Would it in fact be possible to reinstate pluripotency in an intact, fully differentiated cell? And at that time, this was considered a utopian dream by most scientists, or at least that there would be a very complex road to come even close to that goal. But in 2006, after relocating to Kyoto University, Shinya Yamanaka published a paper that spellbound the scientific community. Introduction of only 4 transcription factors could convert a differentiated cell to what became known as the iPS cell. And in only 6 years, this remarkable discovery that the differentiated state could be unlocked and converted to iPS cells, that has really taken the scientific world by storm. And the technology is now used in thousands of laboratories worldwide, including several at this university, and it's used to address important questions, for example, to model and understand diseases in completely novel ways. Shinya Yamanaka received his MD from Kobe University in 1987 and a PhD from Osaka City University in 1993. He is currently professor and director for CIRA, the Center for iPS Cell Research and Application in Kyoto, and also senior investigator at the Gladstone Institute in San Francisco. Shinya Yamanaka has received numerous awards, including the Lasker Medical Research Award and the Wolf Prize in 2009. He was elected member of the National Academy of Sciences in the United States in 2012. And with this as a background, please join me in welcoming Shinya Yamanaka, who will present his Nobel Lecture under the title, Induction of Pluripotency by Defined Factors. Please, Shinya.
Well, thank you very much, Professor Urban Lendahl, for your very kind introduction. It is a great honor for me to be selected as one of the laureates of the Nobel Prize in Physiology or Medicine this year. I am grateful to the Nobel Foundation and Karolinska Institute for having chosen me as one of laureates. Actually, on October 7th, just one day before the announcement, I met the president of Karolinska Institute, Professor Wolfgang Henriksson, in Kyoto. She chaired a session which I attended. When I said goodbye to her after the session, I thought she winked at me. I was not sure. But I'm sure now she did wink at me. So I feel especially honored to share this award with Surgeon Gurdon because Professor Gurdon initiated the field of nuclear reprogramming. Without his seminal work done around the year I was born, I could have never been here today. So thank you very much, John, and I respect you a lot for your brilliant head, both inside and outside. So in my talk today, I'd like to talk on 3 topics. First, I'd like to introduce to you my early days in science as a scientist. Then I'd like to discuss about the researches of my own and by others that led to the generation of iPS cells. Finally, I'd like to discuss the potential of iPS cells. So let me start with this topic, my early days in science. I have to say I was extremely lucky in 2 ways when I was a graduate student and I was a postdoctoral fellow. The first luck was that I had multiple opportunities to observe totally unexpected results that brought me to completely new projects. The other luck was that I was able to work under 2 great mentors. So in the next few minutes, I would like to share with you my experiences when I was a student and a postdoc. So as you heard from Professor Lendahl, I started my career as an orthopedic surgeon. But within a year or 2, I realized that I was not talented at all in surgery. I also realized that even a talented surgeon, talented doctor, cannot help many patients suffering from intractable diseases and injuries. Because of those 2 reasons, I decided to change my career from a surgeon to a scientist. I entered a graduate school in Osaka City University, and I majored in pharmacology. I studied the regulation of blood pressure, mainly in dogs. The mentor at that time was Dr. Katsuyuki Miura. So this is the very first experiment I performed as a graduate student in Osaka City University. We knew that the intravenous injection of a vasoactive molecule, platelet activating factor, PAF, into dogs caused a transient decrease in blood pressure, transient hypotension. Dr. Miura hypothesized that this transient decrease in blood pressure is mediated by another molecule, thromboxane A2. So he asked me to prove his hypothesis. The experiment was very simple. I pretreated dogs with an inhibitor of thromboxane A2. If Dr. Miura's hypothesis is correct, this is what we should observe. So we shouldn't observe any decrease in blood pressure if Dr. Miura's hypothesis is correct. So this experiment was supposed to be a very simple experiment, suitable for a beginner, suitable for a failed surgeon. But what I observed was something totally unexpected. In the beginning, it looks like the thromboxane A2 inhibitor showed any effect. So I observed a normal pattern of hypotension. That's kind of boring result. But after a few minutes, I observed this kind of very profound and prolonged decrease in blood pressure. When I looked at this, I got so excited that I rushed into Dr. Miura's office and take him to the doc. Although this result was totally against his hypothesis, Dr. Miura got also very excited. So for the following 2 years, I examined the the mechanisms of this unexpected result. And that became my thesis after 2 years. So then I decided to continue my training as a scientist in the United States. I became a postdoc at Gurdon Institute of Cardiovascular Diseases in San Francisco. My mentor at that time was Dr. Thomas Inenarity. This is Dr. Inenarity's hypothesis. He was very interested in one gene, ApoBec1, which he thought important in cholesterol regulation. He hypothesized that forced expression of ApoBec1, this gene, in liver would lower plasma cholesterol level. So by using ApoBec1, he thought he may be able to prevent atherosclerosis. So he asked me to prove his hypothesis by making transgenic mice overexpressing ApoBec1 in liver. So I worked very hard, day and night, and I was able to generate transgenic mice expressing high amount of ApoBec1 in a liver-specific manner. One day in early morning, a technician working with me rushed into me. She was taking care of these mice. And she said something strange to me. She said, "Shinya, your mice, many of your mice are pregnant." But they are male. So I got confused. So I went to the animal facility to check the mice, and I found she was right. Many of my mice, both male and female, they looked like they are pregnant. So I further looked into what was going on. And I found this totally unexpected result. It was not babies. It was this huge liver cancers. So it turned out that this gene, ApoBec1, is a very potent oncogene. We can never use this in treating atherosclerosis. It would be a crime. So very naturally, Dr. Inenarity got disappointed. But I got very, very excited. And in a sense, Tom Inenarity got also excited too. He encouraged me to continue working on this gene, ApoBec1. So I became only one researcher who worked on liver cancer in Gurdon Institute of Cardiovascular Diseases. But again, Dr. Inerarity kept encouraging me, kept helping me. So I'm very, very grateful to him. So again, in a sense, I had two types of great teachers, great mentors in my early days as a scientist. The one great teacher is a real teacher, Dr. Miura and Dr. Inerarity, who encouraged me despite the results which were against their own hypothesis. The other great teacher of mine is nature itself, who gave me totally unexpected results and who brought me to completely new projects. I have been trying to be a good mentor as Dr. Miura and Dr. Inerarity, but I found it's very difficult. But I will do my best. So let me move on to the second topic of my talk today. I would like to discuss about researches of myself and by others that led to iPS cells. So my own research that led to iPS cells has started from this unexpected result, from this unexpected liver cancer. Actually, Ian Boren is here with me. He is also a postdoc of Tom Inerarity, so he must know some of this unexpected result. So I try to understand the cause of this unexpected cancer, tumors. And I was able to identify one new gene, which I designated NAT1, Novel ApoBec1 target number 1. Very straightforward name. So ApoBec1 is an enzyme. I identified NAT1 as a new substrate, new target of ApoBec1. And from its sequence, I predicted that NAT1 function as a tumor suppressor gene. And NAT1, I thought NAT1 is responsible for the tumor, for the cancer I observed in ApoBec1 transgenic mice. So I generated knockout mice and also knockout ES cells to prove my own hypothesis. But here again, I obtained totally unexpected result. It turned out that NAT1 is essential for pluripotency of embryonic stem cells, ES cells. While I was working on NAT1, I had to go back to Japan. I really wanted to stay at Gladstone. I really wanted to stay in the States, in the United States. But because of various issues, I had to go back to Japan. But before talking about my life in Japan, let me discuss about embryonic stem cells. As all you know, embryonic stem cells, ES cells, are first derived from mouse embryos in 1981 by Dr. Sir Martin Evans and also by Dr. Gail Martin. ES cells have two important properties. The first one is their rapid proliferation, is their Oh my God. I promise I didn't do this on purpose. Immortality. The other important, I'm sorry for ES cells. The other important property is pluripotency, their ability to differentiate into virtually all types of somatic cells and germ cells that exist in our body. And it turned out that NAT1 is essential for pluripotency. When I generated NAT1 knockout ES cells, they can proliferate normally, but they can no longer differentiate. So NAT1 is essential for pluripotency. This unexpected results again changed my project from cancer to stem cells to ES cells. But again, when I was working on NAT1, when I was working on NAT1 deficient ES cells, I had to go back to Japan, and I suffered from a disease which I designated PAD. Well, you may wonder what it is, PAD. Actually, I coined the word. PAD stands for Post-American Depression. So, I had such a great time in San Francisco at Gladstone in many ways. After coming back to Japan, I was depressed. I was very badly depressed that I was about to quit from science. But very luckily, two events happened in my life that rescued me from PAD. The first event was the generation of human ES cells by Dr. Jamie Thompson in Wisconsin. Before his success, we only had mouse embryonic stem cells. I was just using mouse embryonic stem cells. I was often told by my colleagues, Shinya, that mouse cells may be interesting, but you should do something more related to human disease, human medicine. So that was one reason for my PhD. But because of his success, it turned out that ES cells are very related to human medicine. Because human ES cells share the two important properties with mouse ES cells. We can expand human ES cells as much as we want, and then we can make various types of human somatic cells from human ES cells, such as dopaminergic neuron, neural stem cells, cardiac cells, and so on. Then we should use these human cells to treat patients suffering from various diseases and injuries, such as Parkinson's disease and spinal cord injuries, and so on. So again, it turned out that ES cells themselves could help patients, not mouse patients, but human patients. So when I first read the science paper by Dr. Jamie Thompson, I got so excited. I still remember that moment. The second event, which rescued me from PAD, was my promotion to NARA. In 1999, I became a principal investigator in Nara Institute of Science and Technology. So I had my own laboratory for the first time when I was 37 years old. As you can see here, this institute has a beautiful campus. They have many good faculty members. They have many good equipments. They have many, not many, but some technicians who can help me with my mice. And the most importantly, they have many talented graduate students. So because of these two events, I was able to overcome my PhD, and I was able to continue my science. Because I got my own laboratory for the first time, I thought I should make a clear vision or a long-term goal of my own laboratory. And I made this as the long-term goal or vision of my lab. I want to make ES-like stem cells, not from embryos, but from somatic cells, from patients' own cells. By doing this, we should be able to overcome the moral, ethical hurdle of human ES cells. Because we have to use human embryos to generate human ES cells, many people are against the usage of human ES cells, against the generation of human ES cells and the usage of human ES cells. But if we can make ES-like cells from adult patients' own somatic cells, we can avoid this hurdle, moral hurdle. I thought, at least in theory, we can do this. We can convert adult cells back into the embryonic state because of two scientific findings reported by previous researchers, two scientific streams established by previous researchers. The first scientific stream is, of course, nuclear reprogramming, started by Sir John Gurdon. As you heard from his talk, Sir John Gurdon clearly demonstrated that somatic cells can be reprogrammed back into the embryonic state. The other scientific stream is factor-mediated cell fate conversion, which was initiated by Dr. Harold Weintraub, who showed that a single transcription factor, MyoD, can convert fibroblasts into muscles in mice. So because of these two scientific streams, I thought there should be factors that can reprogram somatic cells back into the embryonic state. By identifying such reprogramming factors, we should be able to make ES-like stem cells directly from somatic cells. However, I had no idea how many factors were required. It could be just one, it could be 10, it could be 100, or even more. So I had no idea how long it would take to identify these reprogramming factors. It could take 10 years, 20 years, 30 years, or even longer. But very luckily, it didn't take that long. It took us 6 years before we identified these 4 factors. In 2006, we were able to report this finding by introducing the combination of only 4 transcription factors, OX34, SOX2, CMYC, and KL4, into mouse skin fibroblasts, we can actually convert somatic cells into ES-like stem cells, which I designated induced pluripotent stem cells, iPS cells. In the following year, 2007, we and others have shown that the same factor combination can make human iPS cells. So I want to use just one slide to show you how we identified those four factors. First, we collected 24 candidate factors with great help of my students and technicians. Among them, these 3 members of my laboratory did essential work. We also established a simple and sensitive assay system to evaluate these 24 candidate factors. 2 members of my lab, Yoshimi Tokuzawa and Tomoko Itsaka, did this work. Actually, we generated knockout mice and knockout ES cells of a gene which I thought essential in mouse ES cells and in mouse early development. However, nothing happened to those knockout mice and knockout ES cells. Those knockout mice were just happy. Knockout ES cells were just happy. Only my student, Yoshimi, became unhappy. But it turned out these mice provided us a very useful system, assay system, to evaluate these 24 candidates. So this is another lesson. We should never give up. Then, by using this simple assay system, very sensitive assay system, another member of my lab, Kazu Takahashi, he did a remarkable job to identify the 4 factors out of the 24. So it was indeed these 3 young researchers who generated iPS cells. Yoshimi Tokuzawa and Kazutoshi Takahashi were my very first graduate students. Tomoko Itsaka is my first technician. Without devoted work done by these 3, we could have never generated iPS cells, at least in my own laboratory. So I'm very, very grateful to these young researchers. So finally, I'd like to discuss about potential of iPS cells. There are 2 major medical and pharmaceutical applications of iPS cells. The first one is pharmaceutical applications. By generating iPS cells from patients, we should be able to develop disease models. Then we can perform drug screening. So this is a pharmaceutical in vitro application of iPS cells. The other application is in vivo application, is cell therapy or regenerative medicine. So let me first discuss about pharmaceutical application of iPS cells by using motor neuron disease. As you know, motor neuron is a specific type of nerve that transmits the signal from the brain to our muscles. in these patients suffering from motor neuron disease, their motor neurons get degenerated and eventually die. So they will become unable to move. They cannot use their muscles. However, they can still think, they can still see, they can hear, but they become unable to move. And in the end, they become unable to breathe. So it's a very, very sad situation for patients and for their family members. Despite numerous efforts by numerous scientists, we still don't have effective drugs for these patients suffering from motor neuron disease, primarily because we don't have a good disease model to perform drug screening. For many diseases, we have animal models by which we can perform drug screening, and by which we have identified many drugs which are effective on both animal models and on human patients. We do have mouse models for motor neuron disease like ALS. Many drugs have been developed using the animal models. Many drugs have found to be effective on mice. But very sadly, all of those medicine drugs turned out to be not effective on human patients. So for motor neuron disease, for people, we do have to use human cells to perform drug screening. However, it is impossible to obtain sufficient amount of motor neurons from patients by biopsy to perform drug screening. So that has been the limitation. But now, with this technology, iPS cell technology, many researchers all over the places in the world, they have been generating iPS cells from patients suffering from motor neuron diseases and other intractable diseases. Then they have succeeded in making a large amount of motor neurons having the same genetic information with patients. Then, now we have an opportunity to generate disease models using patients' own motor neurons. And we now have a great opportunity to perform drug screening using patients' own cells. One of my colleagues in Kyoto, Dr. Haruhisa Inoue, he has generated iPS cells from both patients, like ALS and other motor neuron diseases, as well as healthy control individuals. As you know, motor neurons and other neurons have these kinds of projections that are necessary for signal transduction from brain to muscles. Dr. Inoue have shown that motor neurons from patients have significantly shorter projections than control motor neurons. So we are now using this phenotype to perform drug screening. We have to screen thousands of drug candidates. So we have been using this kind of robotic system to identify drug candidates for patients suffering from motor neuron disease and other intractable diseases. Let me move on to the other application, namely cell therapy, or also known as regenerative medicine. So just like human ES cells, we can expand the number of human iPS cells as much as we want. Then we can make a large amount of various types of human cells from iPS cells, including dopaminergic neurons, retinal cells, cardiac cells, neuroprogenitor cells, platelet, then we should be able to use these cells to treat patients suffering from Parkinson's disease, macular degeneration, cardiac failure, spinal cord injury, and platelet deficiency. In Japan and in some other countries, researchers are conducting preclinical studies to prove the efficacy and safeness of these iPS cell-mediated cell therapy. Next Monday, December 10th, I'm going to receive the Nobel Prize on behalf of many researchers, scientists, who have contributed to the generation of iPS cells and who have contributed to the very rapid progress of this technology. As I described, iPS cells became possible with the three pre-existing scientific streams. The first stream is nuclear reprogramming. Again, it was initiated by Dr. Gurdon, Surgeon Gurdon. Then, Dr. Sir Ian Wilmot succeeded in somatic cloning in mammals, in sheep for the first time in 1997. In 2001, Dr. Takashi Tada demonstrated that mouse ES cells have reprogramming factors. So this stream, scientific stream, was essential for me to initiate our work. The second important stream is again factor-mediated cell-fate conversion initiated by Dr. Weintraub, who unfortunately passed away when he was still very young. The third essential stream is ES cells, embryonic stem cells, initiated by Sir Martin Evans in 1981. Dr. Austin Smith and others have been identifying many factors that play important roles in pluripotency, including OX34, SOX2. In 1998, Dr. Thompson succeeded in generating human embryonic stem cells. So all of these works were essential, were indispensable for the generation of iPS cells. Now, from iPS cells, new scientific streams have already emerged. In 2007, Dr. Rudolf Jenisch provided the first proof of concept of iPS cell-based cell therapy in mouse models. In 2009, Dr. George Daly and Dr. Kevin Egan generated iPS cells from patients for the first time. I have to mention here that there is another stream which has emerged from iPS cells. In 2008, Dr. Douglas Melton showed that cell fates can be directly converted in vivo in living mice by introducing just a small number of transcription factors. This method is now called direct reprogramming, which has been followed by many researchers. More recently, in 2012, this year, Dr. Deepak Srivastava provided the first proof of concept of therapy based on direct reprogramming in mice. So I really hope that in the very near future, these technologies will help patients, either in cell therapy or in drug discovery. I also hope that some of these researchers will receive another Nobel Prize for their works in the near future. So finally, I would like to thank many of my colleagues, of my fellows in two places. The first place is in Kyoto, Center for iPSL Research and Application, established 2 years ago. We now have more than 250 people working in this building. I'm grateful to all of them for their hard work. I am also grateful to Kyoto University for their continuous supports to our institute and to our science. Our president, Dr. Hiroshi Matsumoto, is with us today. Let me also thank the Gurdon Institute in San Francisco. Since 2007, I have a small group, in addition to Kyoto, at Gurdon in San Francisco. Compared to Kyoto, it's a very small group, but they are all talented, brilliant, and independent people. So I visit Gurdon almost every 4 or 5 or 6 weeks. My regular visit to San Francisco, to Gurdon, always activates my science. So I would like to thank Dr. Bob Maley and also Dr. Deepak Srivastava, who are also with us today, for this great opportunity. Finally, also with me today are my family members. I have my wife, my mother-in-law, my daughters, my brother-in-law, my sister. Well, my own mother is in Stockholm, but she cannot make to this hall today. Well, she said, Shinya, your English is terrible, so I won't understand. So instead. Well, as you know, science, being a scientist, is full of joys. But at the same time, it's full of stress. So without continuous support from my family, I could have never been here today. So I'm very, very grateful to my family. Well, I miss my father, who passed away 25 years ago, and my father-in-law, who passed away earlier this year. I believe they are enjoying this moment together somewhere. Well, it was my father who talked me into medicine. And it was my father-in-law who showed me how a doctor should be. My father-in-law was a doctor for a long time. So I really need to bring our technology, iPSL technology, into clinics, into patients. I really have to do that before I meet both of my fathers in the near future. So, thank you very much for your kind attention. Tusen tack. Thank you.
Before concluding the Nobel Lectures for 2012, I would now like to ask both the Nobel laureates to step forward, please. Please, Dr. Gurdon, Dr. Where do we have Dr. Yamanaka? Oh, there you are. And a big hand. Well, thank you very much for those outstanding lectures. And now, can I ask everybody to take your seats? Because I would like you to stay in your seats and let our honorary guests leave first. And thereafter, everybody can leave. So thank you very much for your cooperation here. So let us have the guests of honors leaving first, and then the rest of us can leave. Thank you very much.